monthly Meet the Scientist webinar. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host and moderator for today's webinar. Today, Dr. Dolores Malaspina will present a webinar entitled Identifying Risk Factors and Protective Pathways for Schizophrenia. The Brain and Behavior Research Foundation is dedicated to funding research around the world to identify the causes, improve treatments, and ultimately develop cures and preventative techniques for mental illness. The Foundation has awarded approximately $310 million in research grants over the past 27 years. I'm delighted to introduce Dr. Dolores Malaspina. Dr. Malaspina is the Anita Steckler and Joseph Steckler Professor of Psychiatry at New York University Langone Medical Center and the Director of the Institute for Social and Psychiatric Initiatives. Dr. Malaspina is a 4-time NARSAD grantee and has authored more than 200 scientific papers. She also co-hosts a weekly radio show for the Sirius XM satellite radio channel on psychiatry for Dr. Radio. And I'm happy to say that I had the privilege of being a guest on Dr. Malaspina's radio show, and it's a great show. Today's event will be an interactive event. We'll start with Dr. Malaspina's presentation, followed by a question and answer period. To submit your questions, please use the Questions tab of the webinar control panel. You can submit your questions throughout the presentation. As your moderator, I will present your questions to Dr. Malaspina, and we will address as many as possible. And now, I'm pleased to present Dr. Dolores Malaspina. Dolores, the stage is yours.
Thank you so much, Jeff. It's really an honor and a pleasure to be able to speak in general about research in this very severe mental illness, but also to reflect the impact that funding from the Foundation has had on allowing me to pursue ideas that might not have been very much appreciated at their onset. So without further ado, let me discuss some risk factors and protective pathways for schizophrenia. Now, we know schizophrenia as a condition that involves psychosis, disorganized thinking, loss of drive, emotional deficits, and a decline in function that most often has its onset in early adulthood. There are some new research findings that color the introduction that so many scientists make about schizophrenia. First, we know that males actually have a greater risk of the disease and an earlier onset. Even though females can be very severely affected, males and females are not equally likely to develop schizophrenia. We also know that there is substantial overlap with schizoaffective disorder and psychotic mood disorders, and that occurs with respect to the risk genes, to declining function for many cases, for the treatment and the symptoms. We know that over time, what appears to be a psychotic disorder from a mood may resemble schizophrenia, and that the actual diagnosis can change. We also know that although it is a familial disorder, which I'll discuss, only 20% of people with severe psychotic conditions have a family history. Furthermore, we know there's an increased risk in immigrants, in those with urban birth, and certain other stressors that I will discuss with you. So schizophrenia can no longer be said to affect 1% of the population everywhere in the world. As we're learning more about the risk for disease, we can refine those risk estimates. So what is schizophrenia? This is the cartoon of the blind men describing the elephant to one another, and all together they probably are describing the same beast, but some will focus on the tail there or the trunk. That's the way it is in schizophrenia research. We are so fortunate to have focused scientists who can zoom in on particular aspects of the disease, what it looks like, how it unfolds, the course, the optimum treatments. But then we also have other perspectives that have come to be called a translational research perspective. Translational research is research that cuts across boundaries. It cuts, again, across classic domains for studying diseases. It may go from benchtop to bedside, as we say, but also takes clues from epidemiology. Putting these different types of research information together and coming up with an understanding of the disease is translational research. And that is the domain in which I have found the most intellectual excitement. But of course, you can't do translational research without partners who intensely focus on the individual aspects of the disease and its risk pathways. So the risk pathways associated with schizophrenia are in three large areas. One is exposures, and I show that $sign there. You can wonder, does that mean reward or does that mean stress? And it will vary for us over time. But each of those aspects of the environment and of the brain's response can play some role. And we entered the genetic era. And indeed, we have been discovering and finding genes for schizophrenia. But at the same time, increasingly, we see how development from fetal life forward can influence the adult brain. And the largest and most comprehensive picture of schizophrenia and related psychoses are those that consider each of these aspects. One may predominate more in one person, whereas another person, may have a disease that's based on all of these or an interaction between two of these types of exposures. Now, I don't want to go much further before I make it very clear to you that schizophrenia is not a single disease. It is, in fact, a syndrome. Other syndromes in medicine might include paralysis or congestive heart failure. Now, schizophrenia is a syndrome. That means that there are very many component causes of the disease. And in fact, probably very few individuals have the identical dysfunction. They overlap in causing a problem in a certain brain network, but may do so for a number of different reasons. We know that we can think in terms of the etiologies, and that's what I'm focusing on today. And those include, inherited genes that may run in a family, or large excess copies of certain runs of genes, which we call a copy number variation. Also, we can look to prenatal exposures or stress sensitivity or later paternal age. These are what we call the risk factors that are related to the eventual onset of the disease. But the disease can look very, very different in different individuals. Psychiatric and psychological experts will say that this is the illness we know as schizophrenia, but people can vary in their onset, their symptom profiles, whether or not they're deteriorating or able to maintain some function. Comorbid anxiety or mania or depression can all be there within schizophrenia and be quite variable. So it is a syndrome, by which I mean almost every individual or group of individuals with families may have a slightly separate condition. Now, here's a list I put together of some factors. You can think of them as experiences or as genes. These are factors that we can say across many, many studies might double or triple the risk of developing schizophrenia. We know that some of these have to do with development. So maternal medical conditions, preeclampsia or diabetes, but also maternal malnutrition or maternal obesity can increase the risk for schizophrenia. Then we can look to experiences the mom might have being exposed to severe war or flood or disaster during the pregnancy. Sometimes conditions at the birth, like a lack of oxygen to the infant as it's being born, or low birth weight or preterm birth, all of those pregnancy-related conditions. But then we can look to childhood and adolescence. We can see that abusing cannabis in the early teen years can double or triple the risk for later schizophrenia or related psychotic condition. We know that traumatic brain injury, particularly for people who are in a family with a lot of schizophrenia risk, can double or triple the risk for the disorder. And then things such as I've mentioned before, being born in an urban area instead of a rural area can increase your risk, as can being someone who immigrated from another country or the child of an immigrant. Then we have the genetics, about which I will say more. But before I go further, let me talk about the meaning of risk. So if you've seen any lecture before, or if you keep listening, you will hear me say that on average, perhaps 1 out of 100 people have schizophrenia. So if I wanted to explain to you the meaning of tripling the risk for schizophrenia, I'm going to do it with this example. So out of 100 people who don't have a particular risk factor, we're going to say 1 out of 100 will develop the disease. So suppose I'm thinking about early pregnancy exposure to a very, very severe trauma, and I tell you that that will triple the risk. Or I tell you that a mom having influenza or a severe respiratory infection in early pregnancy will triple the risk. That sounds very frightening to many people. But what does it actually mean? It means that 3 out of 100 people who had that exposure might develop schizophrenia. Well, the most important thing I want you to remember when I say that a factor triples the risk is what it means about people who've had that exposure but don't develop the illness. So something may triple the risk, and for scientists, that's extraordinarily exciting. It's an amazing clue, but if you're an individual who had flu in early pregnancy, you might think, well, what is the risk to my child, and what is the chance of not having the disease? So tripling risk means that 97 out of 100 people who had that exposure would not develop schizophrenia. This is really important because most of our disease genes also triple the risk for the condition, which means that most people carrying those genes would not develop the illness. Now, going back to families, these are a summary of data that were collected over 100 years. It was back in the very, very last century, the early 1900s, when it was clearly observed that people with schizophrenia could sometimes have a taint of the illness within their relatives. And the many family studies showed that if you began with somebody who had schizophrenia, in fact, the risk to their relatives could increase. Although 1 out of 100 people generally in the population had schizophrenia, what we saw was that the sibling, their risk was increased 9-fold to 9%. Now you're all thinking, gee, that means 91% of siblings won't have schizophrenia, and that's exactly right. But the most intriguing thing that we return to again and again is looking at identical twins, because identical twins share all of their genes, but they are concordant only 50% of the time. And that remains both the most compelling information for doing genetic studies, because the risk is increased 50-fold, but the most compelling reason as well to know that there are indeed protective factors and risk factors that do transcend the genes. Now, the list of genes related to schizophrenia keeps growing and growing. When I was involved in my first genetic study, which was the NIH Genetics Initiative, I must say we were expecting a very few number of genes to cause schizophrenia. But that list keeps growing and growing. But each of the genes we find is of a pretty small effect compared to the whole population of people who have the disease. So what we need are larger and larger and larger studies to be able to identify the risk genes. So in the past, we thought perhaps we needed hundreds of people to study to find the genetic pathways for schizophrenia. Now we think we probably need 100,000 people to be able to dissect the risk pathways and what they mean. Finding the risk genes is still one of our best opportunities to understand the pathology of the disease and how to find cures. The genes we keep finding are very often genes related to how cells signal to one another, how cells divide, to the development of the brain, but also to inflammation and to immune pathways. And that immune pathways includes our major histocompatibility loci. So we see that there are many, many genes that are unfolding. Well, a dozen years ago, my group pondered the question, when we were only expecting a few genes for schizophrenia, how could the genes persist in the population when people who have schizophrenia are far less likely to have children than other people? And so we entertained the idea that new mutations may be occurring, and that those new mutations could be increasing the risk for schizophrenia, even though the individuals with the disease might be less likely to have offspring. Now, new mutations had been proposed a very long time ago for schizophrenia, but it was thought at the time that human mutation rates couldn't be possibly high enough to account for such a common disease as schizophrenia. New data shows that, in fact, mutations are quite common in humans, at least as much so as in other species, and that the source of most de novo mutations in humans occurs in the male parent. And that's because the spermatogonia that give rise to the sperm are constantly dividing 200 times and then on 600 times. So with each set of years that a man has lived longer, the cells have divided more times, just like the somatic cells, the age freckles that maybe some of us are getting that occur as the skin cells divide and divide. Cells that divide will accumulate errors. Now, I've shown in this little figure the origin of a mutation as that first pink cell. And what you see is that over time, that pink cell would be occupying a greater and a greater proportion of the available sperm to be used for reproduction. Now you see that a much older father can still have plenty of the green dot sperm, which are the unmutated ones, but sperm with the mutation may expand as fathers age, particularly if the mutation makes an advantage to cell division. That can sometimes be called a selfish mutation. Selfish because it may be an advantage to the sperm, but a disadvantage to the offspring. Now, by comparison, a woman's eggs have very few divisions, and most of them take place while she herself is a fetus, showing an influence of the grandmother's womb. But it is in the male parent that active cell division is taking place. We wanted to see, could father's age then be a predictor, a linear predictor of the risk for schizophrenia? Now, to do that type of study, you can't just recruit many, many people in, because there will be biases in who comes to your study and who is examined. If you want to do ideal studies of risk factors, you need to identify a whole population that you will study, and that is called a cohort. So you've identified everyone you'll examine before you know who has the disease or who has any particular risk factor. Our cohort is every offspring born in Jerusalem from 1964 to 1976. And in that cohort, we were able to link the records without knowing anyone's personal identification, just linking record numbers that we had through the Ministry of Health to psychiatric registers, also linking that to information on the ages of parents and other sources of data. And when we looked at that for the very first time, where we corrected father's age for mothers and mother's age for fathers, what we saw remarkably was that the age of the mother, shown in these red bars, had very little impact on the final risk for schizophrenia, whereas the age of the father regularly increased the risk for schizophrenia. In our birth cohort in Israel, we could explain a quarter of the whole population risk based on father's age. This has been replicated many, many times in different samples when people do population estimates on the impact of paternal age. It's in the range of 15% to this 25%. And if you look at the cases with no family history, it's an even larger percentage. So this was the data showing that father's age was indeed a risk factor for schizophrenia. Now I'm just showing you here how that might compare to Down syndrome and mother's age. We know Down syndrome isn't a point mutation in a gene, but it's a whole extra chromosome 21. And that may be in mother's age because the cells sit around for a long time and chromosomes can fail to separate. But if you look at age 40, you would see that the risk of schizophrenia to a child of a father at age 40 is of the same magnitude as the risk of Down syndrome to a child of a mother age 40. So there is a public health consequence of this. But also there's an opportunity to learn more about the science of what is producing schizophrenia. In that birth cohort, with the second round of funding from the National Institute of Health, we went back and started working through the psychiatric hospitals in Jerusalem to recruit individuals who were born during the study years, who had schizophrenia but no family history of schizophrenia, and whose mom and dad were alive and willing to participate in our study. That study obtained genetic material from mom and from dad and from the offspring who had the type of schizophrenia I call sporadic, which means no other schizophrenia in the family. By next generation sequencing of all of this DNA from mom and from dad, we were able to see, comparing it to the offspring, that there was a new point mutation 5 times. So we identified in this small study of 12 trios 5 brand new point mutations that each increase the activity in a different cell cycle gene. This is very exciting, and we are pursuing this data, and other groups as well are identifying new point mutations in sporadic cases. But what I want to say to you is that as I've learned more and more and examined the literature, my thought is these point mutations may not be sufficient to explain the very large effect of father's age for so many conditions. Father's age is now linked to bipolar disorder, to autism, to obsessive-compulsive disorder, as well as to schizophrenia. So it is a factor that's influencing the risk for many neuropsychiatric disorders. So I've been talking to you, as do many people, of course, about genes. And the gene is the sequence of the nucleic acids along the line of the DNA, the double helix, and groups of these together define a gene. Do you remember what Watson and Crick said when they first published the double helix? They said, "We have found the secret of life." Well, as science goes forward, what we have learned is that just having a gene is different than expressing that gene. That, in fact, that elegant picture of the double helix looks much more like this fuzzy thing I've pictured under epigenetics. That means that the DNA molecule is loaded with histones and methyl groups and other molecules that turn genes on or turn genes off. So gene expression is regulated by epigenetic mechanisms, even including some bits of RNA running in reverse. So our question becomes, what are the factors that change gene expression in addition to what's a risk gene? Will the future let us turn off only the risk gene or turn on a protective gene? What science suggests is that these epigenetic mechanisms allow the fine-tuning of our behavior and our survival to an anticipated environment. So the epigenetic mechanisms are very crucial, and you might think of them as the place where nature meets nurture. Here's a quick little cartoon. If you learn DNA makes RNA and RNA makes protein, if you put a lot of groups or these little switches, those red dots, which could be methyl groups or histones, you will silence those genes, and then those genes will not be expressed. And this is the welcoming perspective of epigenetics and how they will modify gene expression. Now, it's not just the experiences in your own lifetime that might change gene expression, such as being exposed to a terrible trauma in your life, or a severe infection, or having high nurture. But it turns out that some of the changes that arise in your ancestors can become part of the germ cells they pass on to you. Now, the cartoon I show is how someone made fun of Lamarck a long time ago. And Lamarck thought that the acquired characteristics that benefited survival in one generation might be passed on to the next generation. And although my high school science teacher made fun of the idea, Darwin did not. Darwin allowed a room for such a mechanism. So these are little semi-permanent switches that can change gene expression, maybe for an environment that will only last 1,000 years, without changing the sequence of the DNA, without changing the genes. And this information can be transmitted to descendants, and the epigenetic mechanisms are critical for cell functioning. But unlike the DNA sequence, these mechanisms can change gene expression over development. Now, I want to go back to a study we did a while ago, and the exciting molecular component to this study, will be coming forth soon. But in this study, what we did is we looked at mice, and we looked at mice who were females that had had one litter, and those were crossed with an older daddy mouse and a younger daddy mouse, and we looked at the offspring of those mice in many different behavioral paradigms. And the finding was that the offspring of the older daddy mouse were somehow impaired in a number of behavioral domains. So the question is, was that a point mutation, by which I mean a permanent change in the sequence of the DNA that would be passed on to their future progeny? Or might it have been a change in gene expression? This simple first experiment we did suggested it might be just a change in gene expression, because the offspring of the most impaired mice were themselves without those deficits. So what we conjecture is that, in fact, gene silencing may play a very important role, even though gene mutations might also play a role, that mechanisms that may concentrate certain mutations may also be selecting for certain types of methylation patterns. And we need to pay attention to epigenetic influences. Here, for example, are two studies that were done where a mouse and respectively a white rat that had identical genetic backgrounds were differentially exposed in the womb to certain exposing chemicals. So in the little brown mouse, the diet was altered, and look at the change in the physical appearance. We call that the phenotype of a mouse carrying the identical genes. And in this little white rat, an identical sibling in the womb was differentially exposed to an environmental estrogen. And look at that effect: change in weight, change in insulin resistance, change in abdominal obesity, the identical DNA, but different exposures during development. More evidence that the genes may be important, but so are the factors that change the exposures of those genes to make them more or less expressed. Let me also mention about pregnancy. For human pregnancy, it's a very important time. I showed you how many different risk factors for the disease operate during pregnancy. One way to understand this is the intimate association of pregnancy with stress hormone signaling. In fact, the clock that triggers the birth of the baby can be thought of as the cortisol, the glucocorticoid clock that gets set. And very early in the pregnancy, a lot of signaling can directly go from the mother's circulation about possible stressors to impact the placenta and possibly to change the lifelong physiology of the fetus. We call this fetal programming. And it's known that fetal programming, stress exposure of the fetus for many, many mammals and rodents, is associated with adult changes related to diabetes, hypertension, hyperlipidemia, abdominal obesity, and so also low birth weight that can accompany early fetal adversity is also related to later cardiovascular disease. So is prenatal stress also related to schizophrenia? And in which critical period? And I mentioned these studies as illustrative studies in the field, but agreeing and really setting the stage for understanding the critical period. So we wanted to look at this, and what we chose to study was, of course, the same birth cohort I described to you before. And we went back into the birth cohort, which was done in Jerusalem, and we identified which of the offspring in the whole cohort of 100,000 individuals were in utero, had pregnant moms during the Six-Day War, and in studies funded by Brain Behavior Foundation and /NARSAD, to myself and to Corinne Kleinhaus, we were able to lift out and show that the critical period for schizophrenia was in the 2nd to 3rd month, whereas the critical risk period for mood disorders followed and was somewhat later, with secondary periods of high risk for other domains of mood dysfunction. And both of these papers are published and available. But what we see is that there are critical periods where the exposure to stress may impact physiology in the brain, increasing the risk for later schizophrenia. So moving forward, let me introduce yet another perspective, and that is, might some of the genes that we find associated with schizophrenia be maintained in the human population for other reasons? Now, one of those reasons might be that we have found now that many of our colleagues, friends, families, and ourselves carry Neanderthal DNA, and it might be 15% of our genes. And that would be a population which evolved very separately from modern humans. Despite that, what we find is amazing genetic identity, but very different patterns of gene expression. So one possibility is that some human diseases are the result of this intermingling, such that modern humans share genes from the Neanderthals. And we have to stop calling them Neanderthals with any pejorative, because in fact it's likely that they greatly enriched what we otherwise had considered the human population. And that might be that coming together of the two lines, a source of change in how genes are expressed. Another possibility that I put out for you is that it's quite unfortunate that some people develop, of course, severe psychotic disorders, and they may be individuals in whom a number of different genes have come together that greatly impair the functioning of those individuals and reduce the fertility of those individuals. But perhaps it's worth the cost to the entire group of keeping those genes in the population, because a number of studies suggest that perhaps with far too many genes that confer a risk for psychotic disorder, there's a problem, but numbers of those genes may themselves be in some way beneficial to the whole population or to individuals within a group. Now, this is the idea of group selections, and it's quite a hot topic that can be greatly debated in psychiatry and other fields of medicine. But I take the perspective that a person can't simply have reproductive fitness on an individual basis, because humans also rely on the social grouping. So we need to survive for our progeny, not just that they are fit and reproduce, but that the society is one where they can function. So we may very well, in addition to having culture, have genes that impact the structural organization of society. Now, I pointed out to you several times how stress itself may play a role in the risk for schizophrenia. And it's possible that many psychosis-related genes have to do with the impact of stress. And I'll include there with stress, starvation, inflammation, the fighting of infections. Now, of course, I mentioned to you prenatal adversity, and I talked about a war, and that's clearly stressful. In other work that I'm involved in, we've looked at early trauma, childhood adversity, childhood abuse, and found there as well a very strong impact on the risk for schizophrenia. I mean, what might be another perspective on older fathers? Well, it's theoretically plausible, I will put to you, that there may be a way of signaling from a father who's advanced in years that the environment for his offspring might be more stressful, and that that may lead to a programming of the offspring. So it is plausible that many of the effects we see for later paternal age may be a type of programming for a stress, and that may be why maternal adversity may increase the risk for schizophrenia, but also for other psychiatric diseases. And perhaps paternal age is another risk indicator of the possibility of life without dad for the whole developmental period. So I've talked about changes in parents' germ cells, of fetal programming, but along the course of life there are many, many other exposures, some that are risk-inducing, but some that are protective. So how do we understand this profile of vulnerability, but also of resilience? And I would say to you that each of us has a unique developmental trajectory. There are genetic factors in the population which may in general be beneficial, but may in the setting of certain exposures create a disease-related phenotype. We each have a developmental trajectory that interacts with those genes, but we have the possibility that an enriched environment may be very protective. Social support is protective. Early intervention for behavioral abnormalities is very protective. Then there's some exacerbating effects. Immune dysfunction, and the evidence is accumulating that many psychiatric diseases involve a component related to immunity. And then we've got the stress axis and the impact of trauma. Now, you're used to thinking about these things as influencing stress-related disorders and depression, but these same factors show a similar magnitude of effect on the risk for psychosis. Perhaps some different genes, maybe that genetic compartment factor is different, but the other large pathway that we've been paying attention to for depression and anxiety likely operates as a factor for schizophrenia. Now, as I wind up, I say there's reason for good news. We know more and more that there's plasticity in the brain over its whole life. Maybe you've learned that running is good for your brain, and it is, and we can see that in the rodent. We can see that in humans, that there's lifelong neurogenesis, and so that holds out hope for some way of repair in schizophrenia. This happens particularly in the part of the brain called the hippocampus, and you will know perhaps that this is a very important factor in major depression, and that antidepressants themselves are brain growth factors. I mean, we've looked in our own research group and published studies showing that in the hippocampus of people with schizophrenia, where you can use a 7 Tesla MRI, you can actually see decreased thickness of the layer that produces the new brain cells. But I like to also note that 3 of the 4 regions with lifelong making of brain cells are olfactory regions, and olfaction has been a lifelong, career-long pursuit of my interest, and it was the very first grant as a fellow that I had funding for. Don't underestimate olfaction for humans. Here's a picture showing how a research volunteer can sniff out where chocolate's been dragged through a field, and olfaction also plays a strong role in human bonding. So to summarize, we think about the birth phenotype. Can you tell at birth if someone is at risk as an adult? What are the factors in adult that predict disease? Well, we look to the genes you carry. Those genes go back to the history of your population, your ancestry. The gene that might have been adaptive for people over many generations of living at the equator, such as quite dark skin, may not be so adaptive when vitamin D is needed. So the history of the population plays a strong role. But I've told you so do epigenetic changes that control gene expression. Those can be passed on through the environment or just occur at the prenatal time. And to give evolution the benefit of the doubt, these changes that result from fetal adversity are meant to enhance the survival of the offspring. Diabetes, hypertension, doesn't sound like it's survival enhancing, but the brain and the fetus can only use glucose, and hypertension might be there if there was a risk of injury. So things that create the chronic diseases of our population probably evolved as a result of trauma to benefit offspring, at least until they reproduced. So these are some of the sources of my support, lists of my colleagues and collaborators and mentees would be a very long list, but I have extreme gratitude to the Foundation and to NIH and for the possibility to pursue something that is so gratifying. So with that, I will thank you all and thank Jeff and turn the proceedings over to Dr. Borenstein.
Thank you so much, Dolores, for all the work that you've done and for doing such a very clear presentation that was user-friendly for the people following it who are not scientists. We do have time for a couple of questions, and one of the questions that came through that I think is one that probably is on the mind of a lot of people is, are there any universal interventions for prevention that are practical and likely to do the most good?
Well, you know, if I gave a list of the things that are protective against the risk for schizophrenia, you would rapidly see that this is the ideal way that we should take care of the entire population. Although the studies aren't in yet, my advice to future parents is that the father is the one who should be taking the vitamins in the month to year before the pregnancy, because it's actually in the father that all of the cell division is going on in the male germ line. Of course, with pregnancy, the mom needs the vitamins. And that becomes very important. Early and excellent prenatal care is important, and likewise enrichment of the environment. As I say, these are true for all individuals. What might be important, and likewise is true, that at a time of a behavioral problem in a child or adolescent or an adult, whether it's social anxiety disorder, which we see in a third of people who go on to later develop schizophrenia, or in a risk period, that people have evaluations and interventions. Psychological support alone can be very, very neuroprotective. We see that the disease emerges very frequently in the brain resculpting that happens between adolescent and adulthood. Certainly substance abuse is very adverse and increases the risk, whereas having social support and an enriched social environment for a young person is probably protective. Sometimes young people who may be developing depression or who are at risk have a period of social withdrawal. We want to try to intervene so that the social enrichment continues through that period and try to reduce criticisms and stress. A big answer, Jeff.
Really a great answer, because these are the, as you and I have discussed, and as our field is progressing, one of the key issues is the issue of prevention, not just treating something once it happens, and obviously that's important, but prevention, similar to what we do now with cardiac disease by reducing the risk of a heart attack. And you're describing steps that can be taken to reduce the risk of a person developing schizophrenia.
Yes.
Once somebody does develop the diagnosis, so now they have a diagnosis of schizophrenia, what steps should they take to increase the likelihood that they'll have the best prognosis moving forward?
Well, that brings me to a very important point. I think in the U.S., more so than in other countries, there's a great reliance on medications at the expense of psychotherapeutic interventions. And whereas we need the best possible medications and treatments, I think it also greatly enhances long-term outcome to have certain types of psychotherapies. And those may include cognitive behavioral therapies, in which people learn how to manage and function despite some symptoms that might be refractory, and having a relationship with a therapist or with a group, or for some individuals with an institution, so that they feel connected in that way. Now, with respect to the medications, I want to point out that we used to think of schizophrenia in a way called hierarchical, meaning that if we made that diagnosis, we thought all the other symptoms someone had were part of that diagnosis. So someone might be given an antipsychotic medication, and it might be raised to a maximum level. And if someone was still functioning poorly, the thought would be another antipsychotic. I think we now recognize that people who have severe psychotic disorders, whether they're bipolar or psychotic depression or schizophrenia or schizoaffective, and recognizing that these labels may switch over time, we need to be person-specific. Does this person also have depression? We have to look carefully, because some of the symptoms of schizophrenia can make it hard to diagnose depression. But the majority of patients with schizophrenia will have an episode of depression and respond to antidepressant medications. And in the setting where they're continued on their antipsychotic, that does not make the psychosis worse, as people used to believe. The other thing is there a social anxiety component, is there obsessive-compulsive component. So each person needs a careful evaluation for the different domains of symptoms that they have, and then the psychotherapeutic, medication, and other modalities that optimize them uniquely, Jeff.
I think that's a very, very important point that you're making, and it's something that family members and patients need to always keep in mind, that it's not a one-size-fits-all, and that if there's a particular set of symptoms that's still bothering someone, let's say depression, that should be treated, that the person shouldn't suffer in silence just because they have a diagnosis of schizophrenia and are now feeling depressed, but should have the depression treated as well.
Absolutely.
So I think it's very important. One of the questions that came through relates to the point that you made, is perhaps some of the genes for psychiatric conditions may have had benefits for social groups. And
yes.
I'm curious if you could give a little bit more information about that.
So, I mean, this is a very important question. So why might a gene be in the population? Along with that goes the information that we have that most disease genes, for example, even if they increase the risk tenfold, the large majority of the time are not causing disease. There's exceptions, of course. So some people have wondered whether some of the genes, for example, may shift the connections between the hemispheres somewhat, allowing someone to think more holistically or creatively, and that may benefit a whole group. Although if such a person has too many of the different genes that increase the risk, of course, that will not benefit an entire social group. But keeping genes that some people might call creative in the population could be important. We know that some of the genes for schizophrenia increase suspiciousness and vigilance, and clearly too much of that is a danger for the person who goes seeking explanations even for irrelevant things. But having some individuals in a population who are vigilant to the environment would be a way for the group to know that there might be a problem that they should pay attention to. You wouldn't want all members of a group to be surveying the environment for signs of danger. Of course, that would be very stressful, but that may be another benefit. Also, when I first started my genetic research, we thought there were perhaps 150,000 human genes, and that number has dropped to perhaps 20,000 or even fewer. That means that some of the genes that are involved in the brain may play roles in other body systems. So a gene might be a risk gene for schizophrenia, but be very beneficial to some other organ system. For example, rheumatoid arthritis is almost never experienced by people with schizophrenia. 11 different studies have found that each disease seems protective against the other. So might a gene that increases schizophrenia risk be protective against some other disease in a different organ system? We need to think even beyond the brain.
Good, very good explanation, very helpful for people to understand that. In our remaining moments, I think I have a question that many people in the audience are asking, but this is a specific one from a person saying, what, quote, best words of encouragement, unquote, can a parent give their 27-year-old son with schizophrenia? And I think this is something that many people in the audience would really relate to.
Well, you should tell your son and yourself to be hopeful. No, it's more exciting all the time that we will actually find ways to turn genes on and off at the molecular level without the medications that have such sweeping side effects. We know that with nurture and encouragement, even only with the available interventions, people with the disease can have a rewarding life, even if it was not the same life that was imagined before the illness had its onset. I myself remain hopeful despite having in my own family such suffering. We need people who are optimistic, who believe that there's hope in the future, who understand that the mission of the Brain and Behavior Research Foundation itself is to have these new perspectives and to keep hoping and discovering. We know so much more now than when I began my career. So I have seen in real time how the field is being transformed. I encourage other people to be hopeful.
Well, I certainly feel hopeful listening to you. And I want to thank you so much. Your passion comes through in all of the work that you do, and I appreciate it. Your work over the years has had a tremendous impact on our understanding of schizophrenia and will enable new discoveries and treatment strategies to improve the lives of people with schizophrenia. So thank
you so much, Jeff.
And I also want to thank our audience for joining us today. The Foundation, through its research grants, is dedicated to improving the lives of people with a wide variety of psychiatric conditions. I thank you for joining us on that mission. As you all know, the research we fund is made possible through private donations. So if you'd like to make a gift, please visit our website, bbrfoundation.org, or call us at 1-800-829-8289. This webinar has been recorded, so if you've missed any portion of the presentation or would like to share it with a family or friend, visit the webinar page on our website. I hope you'll join us again next month when Dr. Jan Fawcett, Professor of Psychiatry at the University of New Mexico School of Medicine, will speak to us about treatment-resistant mood disorders on Tuesday, August 12th at 2 p.m. Eastern Time. Once again, thank you for joining us and enjoy the rest of the day. Take care, everybody.